To the ATP consumption in the cilia is not surprising because in cilia you have dynein mortar actually driven by the ATP molecule. And then from the result, it found out that if I stimulate the cell with the DC light at different intensity, you will see that all these mean optical density that I put in front of the laser LED light and the highest number of the intensity show that the cell actually suppress the beating results. So it suggests that somehow when you have the biological system that is very stable, that means you should have negative feedback. So I hypothesized that some most of biological systems have at least 2 negative feedback. And when you have more products, somehow you try to suppress to making it. So, and this is the number of the result that I get. So one of the mutants we try, and that the mutant actually lack of the glycolytic enzyme in the cilia. So basically, the lack of one parameter that produces the ATP molecule. And then you see that the standard deviation of the mutant actually lesser than the wild types. So which is possible, support the result. And then when you have some kind of negative feedback, you should be able to somehow break that negative feedback. And so therefore, when I stimulate the cell with the high intensity, not high frequency, and my system is break down. So one of the negative feedback actually pushing down, try to keep the beating down, and one is try to decrease the beating frequency. So this is the evidence of my results, and it's just one of the parameters. So we have so many things. If you have any questions, so you can discuss with me later. Thank you.
I have 1 minute for questions. Any questions?
Why does that imply positive feedback?
Because the system actually, when you say positive feedback, it tries to either increase or decrease in opposite direction. So negative feedback, in my understanding, is that the system tries to be in a stable state. But if it's positive, it's trying to drive the system out of that stable. Thank you.
Okay. Any more questions? Okay, the next speaker is Shiladitya Banerjee from Syracuse University. He's working on, his talk is on activity-induced deformation of cross-linked actin networks.
Yeah. So what I've tried to do here is build a continuum model for actin cytoskeleton. Now, active liquid crystals have been extensively studied but the focus has been on the viscous properties of the gel. So we'd like to know what are the elastic properties of such a network. Now, numerical studies by Dalheimer and group have shown that cross-linked actin networks, where the cross-links are passive, they can actually undergo a transition from an isotropic phase to a nematic elastomer phase. Okay. Now, we'd like to study what is the effect of activity on such nematic elastomers. So, basically, we replace this passive cross-linkers by active cross-linkers. And we build a phenomenological theory using the existing theory of nematic elastomers. Now, the continuum free energy of a nematic elastomer can be broken down into 3 parts. One is the elastic free energy of an incompressible uniaxial elastomer. Another is the usual Frank free energy. Now, the term that plays a role is the coupling between the nematic field and the strain field, which sort of defines the deformation of the elastomer. Now the main assumptions that go into a theory is the incompressibility of the elastomer. We study it in a particular geometry, and we also assume that they are dense, elongated, and stiff crosslinkers so that the anisotropy develop in the network. Now one can actually write down the hydrodynamic equations, and we actually phenomenologically add this active term to the stress tensor. Now this term can be actually justified by more microscopic calculation. To the lowest order in the field, since we are working in a linear theory, this is the most relevant term. So what we see is that starting from a stable ground state, if one includes this term and we solve the hydrodynamic equations, but relatively above this ground state, one sees a Friedrichs-like transition of the pneumatic field. So what one observes is that due to this distortion of the director field, there will be some spontaneous deformation of the elastomer. So what I have shown here is the fundamental mode. One can essentially get various kinds of modes depending on the length of the system. So when activity is actually tuned beyond the threshold alpha C, okay, alpha C depends on the intrinsic properties of the network, like density of. So alpha C actually depends on the shear modulus and the coupling constants of the network, and alpha characterizes the density of active crosslinkers. Another way to see this instability is that the activity can actually destabilize the sound waves. Now, if we had alpha equal to 0, we'll get normal sound waves. But when alpha is tuned beyond alpha C, we get unstable modes. And that might be actually relevant for understanding spontaneous oscillations that are observed in muscle fibers.
Okay, question?
What's the flux density of the ATP hydrolysis needed for this to occur? I mean, how many ATP per centimeter cubed per second?
I'm not actually I'm not actually sure about the numbers presently.
Numbers are important.
Yeah, I know. But we have actually started to work on this model, and don't do.
Your transition implies a splay type of deformation. Is there a possibility for twist or?
Yeah, so actually it will depend on how the director is anchored to the wall. Since we have a anchoring which is like in X direction, we'll have splay deformation. But if it was normally anchored, we'll see other kind.
But no twist.
Huh?
But no twist.
Yeah, because it's essentially one-dimensional, because it is infinite in exterior. So in 2 dimensions, you can't have twist deformation.
One last question.
Are you looking at 2D system or 3D system?
It is like, you can say a quasi one-dimensional geometry. So it is like infinite in x extent, but it is confined between z equal to 0 and z equal to z. So it is not strictly 2-dimensional. So one has a transitional invariance in x direction which plays a role in this kind of distortion.
Thank you. The next speaker is Kelly Burke from Case Western Reserve University and Syracuse Biomaterial Institute. She's talking about liquid crystalline elastomers as soft-shaped memory networks. Hi.
So I'm going to be talking about main chain liquid crystalline elastomers as soft-shaped memory networks. And so in this case, what we're talking about are soft polymeric networks that are chemically cross-linked and that display a physiologically relevant modulus. And so here I have a comparison of human tissues to my main chain liquid crystalline elastomers. And in my schematic, these dots are covalent cross-links, and in between the covalent cross-links are green squiggly lines, which are polydimethylsiloxane, and blue rods, which are mesogens. Now, the siloxane in my networks impart softness, while the mesogens themselves impart shape memory properties. And so if you look at modulus as a function of temperature, you see, first of all, these materials form Smectic C phases, and so there's two transitions actually relevant. There's a glass transition of the mesogens within their Smectic layers. This is not an overall glass transition of the material. And there's also a clearing point, which is the order to disorder transition of the mesogens themselves. And these transitions may be merged or separated based on the mesogen composition. And so there are two actual types of shape memory that are possible in these networks. The first is thermoelasticity, which takes place around the clearing point. And so if you think about a film of material and you heat that above the clearing point and apply a load, and then cool that below the clearing point, this material will elongate. And if you heat it up again, it will contract. This reversible extension and contraction is directly coupled to mesogen order and is related to the stress applied to the material. You can imagine an application of such a material as to be an artificial muscle. These materials also have one-way shape memory properties in which, again, if we consider our film of material, we can heat that above this point, the TG, deform the material, cool it down below its TG, unload it, and you get a fixed, long, elongated shape. If you then heat it up under no load, you have a recovered shape. Though a lot of polymers can do one-way shape memory, there are two things that are important with the LCE. First of all, it's soft at all temperatures, which is unlike other materials. And also, we have near perfect shape fixing and shape recovery. And so an application of this, we're actually looking with a collaborator from the Syracuse Biomaterials Institute, Jay Henderson, and we're looking at these for active cell culture. So what we do is we take flat liquid crystalline elastomers, and through our one-way shape memory process and using a patterned silicone wafer, we can emboss a surface topography on these LCEs. If we then heat them, we can recover a flat shape. And so I have a movie, and hopefully it's going to work. It does. Okay, so here you have your rough shape, and what we're doing is we're heating up, and you'll see that eventually there will be a erasure of the surface topography. So this is important because you think about there's a lot of design variables you have, but what we're moving towards is a next generation type of cell culture material. And with that, I'll take any questions. Thank you.
So the cells are going to be happy at 50 degrees? What's that?
I can move that transition around quite a bit, actually. So that triggering temperature, I can move below room temperature, above room temperature. So that can be tailored, yeah. And the modulus of the material can be tailored as well.
But do cells like temperature changing? I thought cells
Sorry.
cells are finicky and they like just one temperature.
Yeah, that's why a lot of work has gone into making the materials at temperatures relevant for cells. So this one's a little high, but we have other ones that are lower. This one's just convenient for show.
What kind of active cell culture things are talking about?
We're looking at how mechanical stimulations to the cells are going to affect the cell's metabolism, what kind of proteins are expressed, and specifically they're looking at things for cartilage at this point, although it could be applied to other cells, I'm sure.
What kind of interaction the gas can have with the substrate?
With the substrate?
Yeah, what's it?
These are freestanding films right now. So you're just seeing the elastomer. They're pretty, they can be pretty sticky materials, so they can adhere to glass though, and so if you're going to stick them in water, you're not going to have a problem with them slipping off.
Essentially, they would just speak to the substrate.
Excuse me?
So the elastomer will speak to the substrate.
Yeah, if you wanted it to, yeah.
Okay. Next speaker is Valerie Cross from Cornell University, and she's talking about mechanism of cell-cell communication within 3D colloids and matrices during vascular network formation in life.
Thanks. So I am working at Cornell University under Abe Strook, and we are working to understand basically the basic mechanisms that cells, and specifically endothelial cells, use to essentially self-organize from isolated cells and after a period of time into a global connected vascular network. This all takes place in a soft, three-dimensional collagen matrix. So the most basic vasculogenesis in vitro model in 3D is essentially placing endothelial cells in a three-dimensional soft matrix, applying a tumor-conditioned media, and we see this global formation. Cells interact and they form this connected network. So we're interested in those local mechanisms. How are they communicating with each other through this soft material? So we and others have seen through live microscopy that during the first 12 to 24 hours of this process, there's very little migration within the three-dimensional matrix. But there does a lot. The main activity is a sort of filipodial protrusion activity. The cells are sending out arms in all different directions. And we're interested in, one, if there's another cell around, are they biased their search in the direction of that cell? And if so, what are those filipodials sensing? Are they just reaching out, and if they contact another cell, then they'll connect? Or is it some sort of mass transfer phenomenon, some gradient? Or is it actual pulling on the matrix and mechanical signaling? And kind of subsets of that, we've been thinking about possible matrix degradation as creating pathways, as well as possible just contact guidance along this fibrous collagen matrix. So how do we study this? Well, right now we're doing live cell imaging of pairs at various separations, and here we have two cells that after 24 hours end up connecting. We look at protrusion dynamics, direction and angle of protrusions over time with respect to a neighboring cell. And we also fluorescently label the collagen and embed microspheres to see how the cells interact with the matrix. So it's a short talk. So I guess briefly, we have seen a bias in protrusion direction if cells are close enough, indicating a 50 micron or so length scale. And we've looked at possible mechanisms, V adherent, VEGF gradients, MMP degradation. But the main thing that we've seen so far, the filipodia definitely travel along fibers, collagen fibers, so we're seeing this contact guidance, and we're working to see if there's also this mass transfer or mechanics mechanism going on by using tricks with permeable boundaries. So with that, I'll take any questions.
You mentioned a tumor-promoting media. I was wondering if are you modeling sort of development of a normal tissue or the formation of a tumor or a combination of both?
So the model is a vasculogenesis model, which is during development, which is basically what happens during development. The only way we really see that is if you add certain factors that tumors do promote, like VEGF and FGF. So it has relevance in both development and in tumor models.
So those cells are not tumor cells, because I mean, tumor cells I work with, they usually migrate rather fast. So. Right.
These are endothelial cells. These are the actual vascular cells that make up the vessels, the capillaries that invade the tumor. So the tumor is secreting factors or is more stiff. Tumors are fibrous, so maybe the vascular cells are, like we said, it could be a combination of what the tumors are secreting or their mechanics. So.
Okay, thank you. So the next speaker is Xiaofan Luo from Syracuse University, and the title of his talk is "You Can Get Blood from a Plastic, a New Self-Healing Hormone System."
So good afternoon. My name is Xiaofan, and today I will show you some of the work I did on bleeding polymers. So this is actually the material that can bleed, and it's a blend of two polymers. The major component is a thermosetting epoxy, and it's blended with a small amount of thermoplastic polymer, and it's a semi-crystalline polymer, polycaprolactone or PCl. And the blend shows a phase-separated morphology where the epoxy thermoset forms all these spheres 50 microns in diameter in a continuous thermoplastic PCO matrix. And we see that all the spheres are interconnected so they can dominate the mechanical properties to make the material both strong and durable. And we term this material a briquettes and mortar composite. And this slide basically shows how the material can bleed. So now we are looking at the surface of the material while it's being heated. And we can clearly observe this liquid formation on the surface of the material. However, the bulk of the material is still a rigid solid. So this process only happens locally or microscopically on the surfaces of the material. So I'm going to skip the rest of the movie, and I want to just briefly discuss the origin of this bleeding process, this phenomena. So if we take a look at specific volume as a function of temperature for both the thermoset and thermoplastic phases, then we know that at room temperature, pretty much nothing can happen. But if we go above the melting temperature of the thermoplastic, what we have is this huge increase in volume of the thermoplastic phase. And because of the phase transition from a solid to a liquid. If we keep raising the temperature, we can have an even larger volume difference due to the relatively large volume expansivity of the liquid thermoplastic phase. So we explored the applications of this bleeding phenomena. So the first thing we did was for the self-healing of the material. So if we have a crack and we can trigger, if the material is damaged with a crack, we can trigger this bleeding, and the crack can be filled and essentially rebounded by recrystallization. And also, we know that the thermoplastic component, PCl, is a very good hot melt adhesive. And once the material is heated, all the surfaces are covered with a thin layer of PCl. So we can use this material as a adhesive material. And it's a very unique adhesive because it has a high degree of structural rigidity at the adhesive state. And also we know the adhesion is from the crystallization of PCl, so we can easily go above its melting temperature to break the adhesive bond. So that renders reversible adhesion to our material. And here you see it bonds to metals and glass, and in both cases you can pretty much reverse the adhesion by simply heating using a heat gun. And that's all I have today, and thank you very much for your attention.
Time for one question.
How much volume change in the solid part?
Oh, yes, that's a very good question. So the epoxy phase, the thermoset phase, has a very high TG. It's above 200. It's well above 200 degrees C. So the volume, there is some volume expansion, but it's relatively small compared to the other phase. So.
Our next speaker is Pat Mather from Syracuse University. Color-changing shape memory polymers.
Okay, we've combined a dye with a semi-crystalline network that exhibits shape memory. The polymer we're working with is much like polyethylene, except it has on every carbon-carbon a double bond for cross-linking. If you look at the elastic modulus versus temperature of the cross-linked material, it has a very convenient melting point from a leathery consistency to a rubbery consistency at about 50 degrees C. And you can play with the material as a rubber above that melting point, impart a deformation, and then recrystallize to fix a temporary shape. And what we did prior to this movie is to take the material and twist it around a capillary tube, fix it by crystallizing, and then in this movie we dunk it into moderately heated water, and you can get a feeling for the time scale of the recovery. If we look at quantifying such a shape memory effect, we can consider as controlled variables the temperature and the applied tensile stress and measure the strain. And what we see with a sequence of 4 loops of the shape memory effect, a stretching in the elastic state, some elongation during crystallization at stress, near perfect fixing, and complete recovery outside of that first case. Now, this can be used for deployable medical devices, which is the primary thing that got us into this, but also for toys, for example. There's an Australian company we're working with on making action figures. That's where the money is. And so the idea with this material is that we have a semi-crystalline network that upon heating is a regular, disordered, cross-linked material that crystallizes to form a percolating crystalline phase that allows us to keep that temporary state until we heat it. So with Chris Vader's group at Case Western, we decided to combine his dyes that have these long alkyl tails in with our polymer at a very low amount, about 0.1 weight %, and they're partially compatible with our polymer. Our polymer is a solvent for that dye in the melted state, but the moment you crystallize, it does not co-crystallize with the crystalline phase, and so it wants to go to the red state. And so the same phenomenon that drives the shape memory can drive the color change. And so when we combined these materials, we found that the shape memory went in sync with the color change. In the high temperature state where the dye exists as a so-called monomer, it fluoresces green. Once we stretched it and cooled back to room temperature, x'omers form and it fluoresces in a red state. However, and this is the fixed state, when we heated it again, recovery happened at the same time as the color change, which I think I'll show you maybe outside of there. Which looks like this. You can see the shape change in synchronicity with the color change. And so we're studying, this is going to move. We're studying this phenomenon quantitatively now as a function of temperature and the color change.
What happens when you pull it out?
The shape doesn't change, but the color goes back. So the color is completely reversible. In this case, it's a one-way shape memory, so the shape doesn't go back, unless you apply a load, in which case the shape will go back as well.
The cycling speed is just thermal.
Just thermal. Yeah. The length scale of the diffusion is nanoscopic, so the color change is in sync with the temperature change.
What would the color changing part be useful aside from the obvious colloids?
Well, the Department of Treasury is interested in this for security of money. So if you have a reversible color change, that's easy. If you can print it on textile, which is one thing we're working on, without the shape change, it's useful for that. Sensors also for packaging, for like FedEx packages or pharmaceuticals, where the color change can tell you if you, in the irreversible case, the highest temperature the material has achieved. So, but I just showed you one version of it.
All right, next speaker is Mohammed from Syracuse University. His talk is on transporting endotoxin polymer by ABC transport.
So what I want to do today is about a recently developed project in our lab. I work for Dr. Mugoliano's research group, and we're trying to explore the way or the mechanism that some endotoxin export, and the machinery is called ABC transporter. So what is a ABC transporter? ABC transporter are membrane-fitted protein nanomachinery that either bring things from out of the cell to in the cell, or from in the cell to out of the cell, and fueled by ATP hydrolysis. And the analyte that can be translated can range from small like metabolites or even polymer as long as DNA or RNA or even polysaccharide, the focus of this talk. Now, ABC transporter actually are found in every living cell, and that ranges from bacteria to humans. In humans, of course, you have physiological role for ABC transporters, but also sometimes some tumor cells are overexpressing multiple resistant that they are all ABC transporters, and also some point mutation that cause this devastating cystic fibrosis disease, and also other immune disease. In bacteria, I'm talking about Pseudomonas aeruginosa, of course, availability, but also involving virulence and pathogenicity. So this Pseudomonas aeruginosa is really opportunistic pathogen that has become resistant to many drugs in the market, and usually it's lethal to some patients, like cystic fibrosis patients. Now, I focus on an ABC transporter in this bacterium that involved in an export of a prerequisite for the endotoxin called polysaccharide. Once polysaccharide is made in the cytosol, it has to go through this bicomponent ABC to the final destination on the cellular cell and become endotoxin. And this ABC transport again fueled by ATP, and we have two subunits, one called WZM and the other WZT. Now, this what we expected from this complex is that this one has to form a hole in the membrane, and this energy driving subunit to interact with this hole or pore. Now, to define the proactivity of WZM, we did some electrophysiology, and this basically shows you that the pore forming activity of WZM, if you separate two chambers with electrolytes, and if you if you protein from a pore, you see an opening. If it doesn't, you don't see an opening. Current is 0. And with many biochemical data, we actually added to the model where the toxic subunit actually a 4 subunit, not 2 subunits, implicating in a more binding sites for polysaccharide to go into this nanopore. And with that, I end this one.
Thank you. Question? Okay, thank you. So our next speaker is David Vesicles from Syracuse University. He's talking about using synthetic nanopores as stochastic sensors.
All right. What I want to give you today is just a flavor of some of my ongoing research and the system I'm building. And let's get started. So what we do, I work with Professor Mogalanu in Syracuse University, and we do single molecule biophysics. What that means is we study proteins, nucleic acids, and polymers one molecule at a time. And this is important because when measuring these molecules in bulk, often they adopt many different states, but in bulk you only measure, or you measure mostly their most probable states. When we probe them in single molecule fashion, we can look at their less common states, which are sometimes important for, say, because they are they bind to drugs and so forth. So I'll just tell you a little bit about how we actually see or how we measure one molecule at a time. And we use the resistive pulse technique. What we basically do is we fill a chamber with a salt solution and have a partition. We create a nanopore, say 5 nanometers in diameter, just a single one between the two salt fats, and then we apply a voltage across there and see an ionic current. So we can measure that ionic current in time. If there are no analytes in the sample, we'll see something like this. That's the y-axis is current, x-axis is time, and we'll see just an open pore with an open current. When an analyte passes in the pore, we'll see a drop in current, because if it occludes some of the ions from moving through, and we can measure that. So the different things we can measure, one molecule at a time, are the amplitude of the drop and the dwell time inside the pore, for instance. So this has been done for a while with biological nanopores, and also it's now being done with solid-state nanopores, and that's what I'm working on now. So the motivation for using solid-state nanopores is that you can tune the size. So with biological nanopores, it's basically you have a fixed size. So we can use different sizes. They're more easily integrated into devices, and we can apply higher voltages. So we can put higher forces on to charged particles within the pore itself. So right now, we've made progress. We can make pores between 2 and 20 nanometers. There's pictures right there. We make them in silicon nitride at Cornell, and they're hydrophobic, so we're having a little difficulty wetting the pores, but we're getting there. So far, we can see blockades due to double-stranded DNA, single-stranded DNA, and we're looking at proteins, so we can see DSA proteins. And that's all the time.
Why do you use silicon nitride?
Well, so we make windows of silicon nitride that are 30 nanometers thick. It's convenient to do so. We put them on a silicon substrate, grow them, and then take away the bottom silicon. We have a nice 30 nanometer thick window. So we drill it with an electron microscope. It's convenient, that's it.
Next speaker is Desmond Ahn from Cornell University. He's talking about crystallization and dislocation dynamics of non-spherical colloidal particles in a 2D environment.
Okay, so hi. So my name is Desmond. I'm an undergraduate freshman at Cornell, working with Sharon and Tai. So today I'll just give you a brief introduction to my work about packing in asymmetric dimer colloids. So packing, the problem of packing is very general. It's how much of an object you can fit to a certain area, into a certain volume. So let's first start with the packing of spheres, or in this case in 2 dimensions. So it's the packing of circles. It's been very well studied, and the most efficient way is hexagonal packing, where each sphere is surrounded by 6 nearest neighbors. So the picture over there is a picture of spherical colloids. Next, why we take the next step up? We go to symmetrical dimers. So a symmetrical dimer, each lobe looks like a sphere itself. So maybe you can expect it. You can expect each individual lobe to pack like spheres, to tile like spheres. And in fact, we find that they do. The individual lobes of the symmetric dimers do tile a triangular lattice, and that's what Sharon is studying. But now, what happens if we break the symmetry and we go to asymmetric dimers? Well, in the first case, if you have a lobe, one lobe which is much smaller than the other lobe, you can expect that that lobe might be able to fit into the interstitial sites of a spherical crystal, and we do observe that. Right, so yeah, this interstitial phase is very interesting, and we plan to study. But then the next question: what about everything in between? So there is a big question mark. These are some of the particles that we have, and you can see that already the crystallization phases are not so trivial. You can observe things such as there's an oblique crystal over here where all of them are aligned. They're aligned parallel to each other. You can see like waves, and it's not trivial, and so I'm trying to understand this. So let's take a look at this picture. So here's the SEM of the particles that we have. This is a G of R plot, so this is the pair correlation function. The peaks show that there is certain long-range order in this system. So we are talking about a crystal. It's not just any random picture. And so in this plot, we track each particle, and if it's surrounded by 6 nearest neighbors, we call it black. So there is some sort of hexagonal or 6-fold symmetry here. So, so that brings me to my conclusion. So as we go from spheres which exhibit hexagonal symmetry, hexagonal packing, to symmetric diamonds where the loads exhibit hexagonal packing. So that we are trying to find out what happens here when it goes from spheres to symmetric diamonds. So we've already seen some of the non-trivial crystallization phases that they exhibit, and it will be interesting to study them. And note that this is only a function of the geometry. It doesn't matter what the particles are made of. And in the future, we can study how the material properties of materials that are shaped like this react to shear. That's all. Any questions? Yes?
Did you say what the particles are made of?
Well, okay, so these particles are just polystyrene spheres. So they are hard spheres, they don't interact with each other. But it doesn't change the packing of the packing is just a matter of the geometry of the particles.
But how are the dimers made with the different aspects?
Oh, how are the dimers made? That's a long story. I can talk to you over tea. Yeah, it won't fit into 3 minutes. Yes?
How do you differentiate your GFR from the GFR like a glass or a liquid?
Sorry?
And why is your GFR different than you would expect for a glass or liquid?
Why is my GFR different from a glass or liquid? Because
for a crystal, I expect it to be much spicier.
Well, maybe it's maybe I don't know. Yeah.
Okay, you might want to discuss.
I can talk to you after that. Thanks.
Okay. Can you ask him your question later? Okay, thank you. You're running out of time.
Yeah.
All right, next speaker is David Quinn from Syracuse University, and his title of his talk is Structure at the Leading Edge.
View.
Good afternoon. So the leading edge of a crawling motile cell is a dense network of actin filaments, which can grow and thus protrude the membrane forward in the direction of motion. And one way the cell proliferates new filaments is to branch these new filaments off of existing filaments in the network. And along with a host of other proteins within the cell, this network can be dramatically remodeled to accomplish any task the cell may need to do. So one question we're interested in is, what does branching do for the system? So two things that a branch can do is it can either grow forward in the direction of the membrane to keep up with the modal edge, or it can reach out to the existing network and connect up, which could provide some sort of stability, elastic stability. And so we've come up with a model, and we modeled the actin network as a directed percolating network, and we measure a few quantities of this network, and I'm going to focus on a couple today. using two of the quantities that we measure, the amount of material that's consumed as the network grows in some time, and the longest filament, which defines the leading edge of the cell in our simulation. And we have this cost function. And here, each dot represents a grown network that was a completely spanned structure. So we're sort of restricting ourselves to only networks that form span networks. So these measurements are dependent on the amount of time the network takes to span. And so here on the horizontal axis, this is the preferred branching angle that we put in by hand and grow the network with. And it ranges from 0 all the way up to 90. And so the measured quantities vary monotonically with the branching angle. That is to say, with the small branching angle, it's, for instance, if I want to reach out to my neighbor, with a small angle, it's very difficult, because all I want to do is grow forward. But if I have a more larger angle, let's say closer to 90 degrees, it's much easier to reach out to my neighbor, so the structure would span more quickly. So hence it's monotonic with the branching angle. And so defining this cost function and then minimizing this cost function, we find that for this type of network, where we just consider branching, we find that asymptotically, we find that there's an optimal angle of roughly 25 degrees. And we've done some simple analytical work to show that under physiological conditions, we can arrive roughly, using the same kind of model, we can arrive roughly analytically with the same value of the angle, 25 degrees. And so with that, I'll leave you with my summary slide, and I will take any questions that you have. Yes?
So there's no force applied anywhere in your simulations?
No.
You're not looking at the network's response to stress?
Not yet. We're working on doing that. Yeah.
One more question?
If I understood right, you said the best angle of 25 degrees. Now up to 3 and 70 degrees. Might there be a reason for it?
Sorry, say that again.
Up to 3, you just write here 70 degrees. Do you see a reason why 70 instead of 25?
No, we don't. Not at this moment. We don't see what the reason is for 70 degrees. In fact, one of the reasons to do this particular project was to see if we could arrive at some sort of geometric reason of why it should be 70 degrees. So there's this. We formulated this geometric competition, and we don't see 70 degrees. So we're still searching for that answer.
Perhaps the neural networks being able to respond to stress is extremely important.
Exactly. That might be so.
Okay, thank you. Our next speaker is Santao Wang from Cornell University. He will talk about self-assembled 2D TMV arrays on fluid lipid membranes.
Hello. This work is done by the Brookhaven last year. So we use the bio-nanoparticles, the building blocks, because they are monodispersed and surface can be easily modified. So to form the 2D structure, we need a 2D confinement and also the lateral mobility. So we use the lipid monolayer, which is fluid, as a template to absorb the nanoparticle to the surface, then form the structure. So here is how we make the samples. First, we have a substrate, and then we coat it with the OTS film, and then we inject the lipid solution. So the lipid is neutral and charged. So then we have a monolayer on the surface. Then we inject the TMV particle, which is tobacco mosaic virus. It's negative charged. So because of the electrostatic interaction, so we have form a 2D arrays. So then we use the X-ray reflectivity to measure to monitor growth of the monolayer. So we find out it's a monolayer of the TMV at the surface. So then we use the GZAX to study the in-plane structure of the surface. So you can see the scattering, in-plane scattering becomes stronger and stronger as time goes after injection. And then from the first order peaks, we can know the spacing, average spacing is about 40 nanometers. So TMV images shows the agrees well with the X-rays. So how we make this arrays more ordered? So we inject the calcium into the buffer. Everything else is keep the constant. So we see the scattering peak. We can see the second order peaks too. So we after calculation for the order positions, so we can see the average spacing is about 20 nanometers, close to the diameter of the TMV rods. So and that shows the alpha images. So this slide shows how important of the lipid monolayer is. Without lipid monolayer, you see the rod is randomly stick onto the surface, but locally maybe have some pneumatic orders. So then we did some charge dependence of the lipid monolayer. So the symbol, each color shows the different charge density on the lipid monolayers. And then how we model the structures. So we assume the rod are parallel within each domain. So as shown here. And then TMV and the FM images agree with this assumption. So by fitting this equation, we have to know the form factor of the rods and also the structure factor. So first, we obtain the form factor by the solution scattering. So by modeling that, we can obtain the form factor of the single TMV rods, and then the structure factor, because it's a liquid. So we model it with the liquid structure factor. So this shows the without calcium structure, and then we see the spacing decreases as the charge density increases, and then that's for the structure with calcium. So you will see the domains increases, and with that I include here. Thank you.
Have you tried different compositions of lipids? I know you have DOTC.
Yeah, DOTC and the DOTAP.
Have you tried different compositions like with different charges? And also, have any of your rods inserted and gone truly in from like maybe before or something else in your liquid?
Yes, I show you that the charge density is when we vary the variation, the ratio between the DOTAP and the DOPC. And also we try different lipids too, like DOPG. We can talk about later.
Quick question.
How do these charge densities compare to the surface charge density of TMV?
It's sometimes low because we vary the charge densities from all charges DOTAP to the no charge DOPC. So.
And is that comparable to the surface charge density of TMV?
TMV charge is around 2,000 electrons on one single, so probably comparable.
Okay, thank you. Our next speaker is Shiliang Xu from Syracuse University. He's talking about a toy spin model of jam solids.
Okay, so today I'm going to talk about some model for jamming. So the jamming transition happens when the packing fraction increases to a critical value, at which point the system becomes amorphous from a fluid-like system. So here I show some simulation results in 2D biodispersed soft-sphere repulsive soft-sphere system. Here, the packing fraction is below the critical value, and here we can see that the packing fraction is slightly above the critical value of the packing fraction. So, and here the blue lines, they are showing the contact force induced by the overlapping between particles in the system. So we can see that the contact force network is heterogeneous, and the system in jam state is disordered in the sense of that the contact network is not a perfect lattice. Besides, we can see that there are some geometrical frustration in the system, and it brings in many local energy minima. In addition, simulation has suggested that some quasi-localized vibrational modes here. As you can see in this right figure, that these vibrational modes, they are not fully extended, but they're kind of weakly coupled into some swirling-like groups. So, well, we want to investigate this kind of phenomena more, since the larger scale simulation turns out to be very difficult. So we want some toy model to explain that. Here's our model. Our model is basically like an XY model with a random phase shift term. But now, in this case, this phase shift term is not random, but depends on the local geometry. That's the coordination number of site I and j. So we can see from this XY version Hamiltonian that if Aij is 0, this coupling is ferromagnetic, while if Aij is pi, then the coupling is antiferromagnetic. So by including both ferromagnetic bonds and antiferromagnetic bonds, we have this whole system frustrated. So the motivation is here. So we think about hexagonal lattice. If we remove one particle here, the particles surrounding it cannot move into fill in this blank without any edge cost in a fully packed situation. So they got stuck. So we think that by removing one particle, we change the coordination number of particles, and then we get some frustration and disorder. And by diluting lattice, we can have uncorrelated disorder or correlated disorder, depending on the algorithm, how we dilute the system. Finally, I show a more specific example here. This is a random graph, and we have these red bonds, they are antiferromagnetic bonds defined in the previous slide. So here, this closed loop with only one antiferro bond, this is frustrated, and this loop with 2 antiferro bonds is unfrustrated. So we conjecture that this kind of toy spin model will, well, in the correlated case, in the uncorrelated case, it's just a spin glass. In the correlated case, we conjecture that it may produce some quasi-localized spin wave. So we are doing both analytical and simulation work on that right now. Thanks.
Okay, we have 3 more talks. Can we leave the question to the break for these last 3 talks, because it's already... Okay, thank you. Our next speaker is Zhangwei Yao from Syracuse University. He's talking about shape of structured vesicles.
My topic is about the shape of structured vesicles. Recently, there are a large structured vesicles synthesized in experiment using diblock copolymer with side chain nematogens, and it looks like this. The size of the vesicle is at the scale of microns, and the size of the building block is at the scale of nanometers. And the whole vesicle is composed of symmetric order on the body, and isotropic bars. It's just like this. This is the symmetric layers. The curved fingers are just symmetric layers, and there are 2 isotropic bars on the 2 ends. And there is a phase boundary between the symmetric order and the isotropic domain. This is the phase boundary. Our research is to understand how the internal structure will influence the shape of the whole vesicle from the aspect of free energy. The free energy of the vesicle is composed of 3 parts. First one is the Planck free energy. The first term, the divergence term, describes the spray deformation of the order. And the second term is the bending term. It describes the bending deformation of the directors. And here the divergence term and the bending term are defined on the curved space, since the vesicle is curved. The second term of the free energy is the coupling between the extrinsic curvature K and the directors on the vesicle. Here, this term describes the bending around the n direction. The n direction is just the direction in this direction. And this term describes the deformation around the t direction. T direction is just this direction around the curved finger. The last part of the free energy is the line tension of the phase boundary. Here, L is the parameter of the phase boundary. Actually, this term can explain the budding phenomena of the vesicle. Actually, the budding phenomena of vesicle can be attributed to the competition between the bending energy and the line tension. That is, here the symmetrical domain will continue to grow until the bending energies and the line tensions are balanced. Thank you.
We have time for one quick question. Okay, so our next speaker is Claire Yu from UCY. She's talking about beating cilia in frog embryos.
Oh, it died. Okay. That's a loose connection. All right, so I want to show you some movies of beating cilia and frog embryos, but let me motivate this by saying that in your body, cilia are very important. You beating cilia throughout your body moves mucus up your windpipe, up your trachea. When you smell, it moves air molecules past your olfactory sensors. You also have mucus beating cilia in the fallopian tubes, moving mucus, and the egg in your brain, the fluid encasing your brain, your spinal column, beating cilia move that. And also, it helps break the symmetry in a developing embryo. Why is your heart on your left side? Turns out beating cilia play a key role in that. In developing embryos, you've got a body plan, and cells need to know which way is up and down and front and back. But how do you ask a cell its sense of direction? Which way does it think it should be pointing? One way to interrogate the cells is through cilia, either which way they point if they're stationary, or which way they beat if they're moving. And so we decided to take a look at some beating cilia on the skin of frog embryos. And basically, this is like a giant forest. You've got a cell which has 100, 200 beating cilia. They're 15 microns high. They're about 1 micron apart. It's like a forest with trees, and the trees are all moving, and how come they don't crash into each other? And I wanted to know the answer to that, so I have some colleagues at Irvine, Peter Taborik and Fawn